Welcome back for part 2 of muscle tissues. Now we're going to look at the exact process of how this cross bridge cycling happens for a muscle contraction. As I had stated earlier in part 1, there's more to this cross bridge cycle than just the myosin heads attaching to the actin and moving it along. There's a little bit more to it. So before any kind of muscle contraction will happen, we have a low amount of calcium available in that cell. We want to keep it that way to prevent any kind of contractions that should not happen. And since the calcium levels are low, remember that tropomyosin is going to block that active site on the actin so that the myosin heads cannot attach. That's what keeps your muscles in a relaxed state. We have voltage sensitive proteins that are in the T-tubules. These kind of help to change the shape. And then that's what causes that sarcoplasmic reticulum to release calcium. So when they get some kind of nerve impulse sent to them, those voltage sensitive proteins, as that depolarization happens, is going to change that shape. That's what's causing these calcium ions to go into the cytosol. And when the calcium is high in that intracellular space, the calcium gets pumped back into the sarcoplasmic reticulum at the end of the contraction. But myosin heads can attach to the actin once that calcium attaches and moves the tropomyosin out of the way. This allows for those myosin heads to connect. Those myosin heads connect to the actin. And that is what forms that cross bridge. And as those myosin heads keep pushing the actin over, it shortens that entire sarcomere, which causes that muscle to contract. So when the nerve stops telling that muscle to contract, that is when that calcium gets pumped back into the sarcoplasmic reticulum and the contraction will end. So here's the 4 steps. The step that's been missing from the parts that we've looked at is the use of ATP. We need ATP to help our muscles contract. ATP is our cell's energy. So for that cross bridge formation, we have our myosin heads. They will attach to those actin filament, those active sites. And those myosin heads pivot and pull that thin filament towards that M line. For the cross bridge to detach, so those myosin heads actually detach from the actin, ATP will come in and attach to that myosin head, and that's what causes the myosin to let go of that actin filament. With that ATP, by moving the head of that myosin head, that hydrolyzes ATP into ADP. And the energy from that reaction is saved in the myosin head for the contraction. So let's look. So when these myosin heads and the thick filaments attach to the actin and the thin filaments, you can kind of see these holes here. This is where that myosin head is going to attach. If you remember from chapter 2, when ATP gets hydrolyzed, it turns into a DP plus a phosphate group. So in this first step, we have our myosin head attaching to our actin. We have ADP plus this phosphate. So what happens next? You see this myosin head went in this direction. It tilted its head. By doing that, it lets go of the ADP and that phosphate. And that's causing your muscle to start to contract. The next step, ATP comes in. ATP binds to that myosin head, and that's what causes the myosin to let go of that actin thin filament. Now it causes that myosin over here to go back to its resting position, cocking its head by hydrolyzing ATP. So as the myosin head goes back to its normal position, this is where ATP gets hydrolyzed. It gets broken down from ATP into ADP plus that phosphate. This now, through that reaction, we took that energy and stored it, this is energy, in these myosin heads for when the next cross bridge is created. And then we go back to this stage where our cross bridges are being formed. So this is kind of the whole picture of the whole sarcomere. If you remember, as these myosin heads attached to this actin, the actin gets pulled this way, and the myosin heads really don't change much. They don't change their positioning much. So rigor mortis. Rigor mortis is what happens after you die. Your muscles get really stiff. And it actually starts about 3 to 4 hours after death. Again, I said our muscles get stiff, and it peaks at about 12 hours. What is happening is we're getting a lot of calcium that is being released out into the cell as these muscle cells are kind of starting to die. But because of that, we have all this ATP that's not being synthesized. So our calcium doesn't get pumped back into that sarcoplasmic reticulum. So our muscles, because this calcium, all this calcium is out there, that calcium removes that troponin that allows that, or attaches to the troponin, which allows that tropomyosin chain to be moved from the binding sites of the actin. That allows those myosin heads to attach, but then they're stuck there because there's no when we're no longer alive, we no longer make ATP. So there's no ATP there to let the myosin heads come off. And because of this, muscles get really stiff, and they can stay that way for days, like 2 to 5 days. And what causes the body and the muscles to finally relax is the breakdown of the proteins in those muscles. When those muscles start to break down, that is when everything starts to relax again. So how does our whole muscle contract? Think your biceps brachii in your arm. How does this entire muscle contract? Well, let's look at it. So we kind of talked about when these neurons come in, they have a bunch of projections that come out, and each one comes to a different muscle fiber. So since we have all these different nerves that are each nerve terminal is attached to a muscle fiber, that allows that entire unit, every muscle fiber that is attached to that one nerve, that unit contracts when that nerve impulse is sent. So when those muscles contract, they're going to produce some kind of tension. That tension is what allows you to pick up your pencil, what allows you to walk, what allows you to do any kind of movement you want. But not all contractions do that. We have what are called isometric and isotonic contractions. An isometric contraction, the muscles don't shorten, but muscle tension increases. But what you're trying to pick up is maybe heavier than you can actually lift. Versus an isotonic contraction, that is where muscles will actually shorten so you can lift something up. So force and duration of the contraction, they're going to vary in response to stimuli of different frequencies and intensities. So how often are these nerves sending action potentials to these muscle fibers determines how strong that contraction is going to be. If it's continually sending, it's going to create more force. We talked about the different muscle fibers coming together. The one nerve that comes to many different muscle fibers, that is technically called a motor unit. That is where that one nerve tells all the fibers that it attaches to contract. That is a motor unit. So as I said, it could be 4 muscle fibers. It could be several hundred muscle fibers, depending on where they're located in the body and what kind of movement we're trying to produce. Is it your eyes to look left and right? Or is it your legs to walk? They all produce they have different size motor units. So that motor unit, when we get that action potential, it does spread throughout the entire muscle, but because if it's coming from one motor unit, stimulation of that one motor unit can only cause kind of a weak contraction within that muscle. It doesn't work as well. But here is what I'm talking about. These nerves coming out of, this is actually your spinal cord. And the nerves that come out, and these are going to all these different muscle fibers throughout. We also have an electron micrograph showing how these nerves just kind of branch out and go to every single muscle fiber. So what is a muscle twitch? A muscle twitch is when only one action potential is sent to that muscle unit and you get one small contraction. If you've ever had a muscle twitch in your body for something for some reason, then you kind of know what I'm talking about. But we get one action potential sent, so there's one short contraction and it's done. That is what a muscle twitch is. And we can actually record and observe muscle twitches on myograms. Myo, muscle, gram, it's a way of monitoring things. So we kind of see a line of contraction activity. So here is one of those myograms. We see where the contraction happens, and where the relaxation happens. We see the period of contraction, the period of relaxation. We see when all of that is taking place on one of these myograms. Just as I said, we have a latent period. That's when the nerve impulses were sent to that muscle. We talked about in part 1 of this, that when that action potential is sent to that muscle fiber, it's not an immediate response. It takes a few milliseconds even for something to happen, but that is that latent period. We don't see that muscle tension yet. We see it when we get to that period of contraction. And that's that crossbridge formation and creating tension. That period of relaxation, that's when all that calcium's getting put back into that sarcoplasmic reticulum because we don't need it. We don't want continued contractions. And as this graph shows, it contracts a lot faster than it'll relax. So there's a lot of things that kind of affect a muscle twitch. How strong is that twitch going to be? How long is that twitch going to be? It could be because of the metabolic properties of that muscle fiber. It could be have to do with those enzymes. Like your eye muscles contract really fast, but very brief. Your calf muscles, they're going to contract a lot slower, but they're going to hold it a lot longer. So as I said, the bigger muscles usually do more. They take longer, they can have stronger contractions than, say, your eyes. There's muscles that go around your eye to help them move. So here is a chart that compares different muscles. The one in blue here, this blue, it goes up and down super quick. That is your lateral rectus. That is in your eye. And when that muscle contracts, it lets you look out laterally. The next one, your gastrocnemius. That gastrocnemius contracts quick and relaxes quicker. That gastrocnemius is in the back of your calf. It is the muscle that you can see that gives your calf its shape. The soleus. That soleus is technically a muscle that is underneath the gastrocnemius. It's bigger. We'll look at it when we're in the lab. But this muscle is a lot bigger. It's going to contract slower. A lot slower than that lateral rectus. So our muscles, they kind of have a graded response. So what that means is not all of your muscles are contracting at the same time. That would cause a lot of fatigue really fast. So depending on what you need to lift something, only some of those muscles are contracting while others are relaxing. So if you're trying to pick up something kind of heavy, some muscles start to contract, but those aren't enough for you to pick up something. So more muscles, more fibers contract, and then more fibers contract until you can pick up something that's really heavy. So those different grades of responses are either due to the changing frequency of the stimulation or the strength of the stimulation. But our muscles technically respond to changes in stimulus frequency. So if you think about it, our muscles respond to electrical communication. We use those sodium, potassium, even that acetylcholine from that nerve cell to that muscle cell to communicate. And it doesn't matter how much sodium comes in, as soon as we start to get positive in that cell, it's going to start closing off those sodium channels and then releasing potassium. So this strength of that stimulation really doesn't make any difference. It's how fast are these impulses, these action potentials coming to a cell or to that muscle cell or that muscle fiber. And I said on the last couple of slides, a single stimulus, one action potential will result in a single contractile response. It's contract, relax. And then that's it. But if we get more than one stimulus sent to a muscle, this is what we would see. Contract, relax, stimulus, contract, relax, stimulus. These are individual twitches, but we're sending one action potential, get a contraction. Send another action potential, get a contraction. Send an action potential, get a contraction. That is what's happening in this picture. But a wave summation or temporal summation, that is when two stimuli are received by that muscle in rapid succession. So it's like boom, boom. One, two action potentials being sent. Action potential, action potential. So muscles can't relax between each stimuli. So those twitches start to increase in force even more with each action potential or stimulus that is sent. That stimuli becomes stronger. Those force of contraction becomes stronger. And that happens because additional calcium is released with that second stimulus. That's what causes your muscles to shorten even more. So here is temporal summation. As this muscle starts to relax, we get another stimulus. Here's our initial stimulus. Oh, here's another one. We haven't relaxed yet. Oh, there's another one. Oh, we haven't relaxed yet. There's another one. So you can kind of see the tension, the strength of that contraction gets higher and higher and higher and higher. This is what produces a strong contraction. So if the number of stimuli being sent, the number of action potentials being sent to that muscle, if it increases, gets faster and we keep getting more and more stimulus going, one after another, after another, after another, that causes that strength and that force. And it can cause us to reach almost maximum where every single muscle fiber is contracting. But these wave summations that are coming through, they help to produce smooth, continuous contractions. If we continue to send stimuli over and over and over and over and over and over and over again, that contraction will eventually progress to a sustained contraction. And it can also, if we hold that sustained contraction long enough, it can cause like a quivering contraction because you're shaking. It's called unfused or incomplete tetanus. But if you've ever held a weight out in your hand with your arm straight, just kind of held it out there as long as you possibly can, you can hold it out there for some time before your arms are going to start shaking because it's going to keep recruiting more muscle fibers to help hold that weight up until all the muscle fibers are being used. And then it's going to eventually cause your arms to start shaking. And this is kind of what it looks like. We get stimulus, 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 stimulus. Oh, no more, we can relax. So this is where we're holding a weight out. Say shoulder height out to your side. You're just holding it here. That is what allows this is what you're doing that could be causing these constant stimuli to being sent to keep contracting the muscle, keep causing more fibers to contract until all of them contract. So if that stimuli reaches its maximum, there is a maximum point in there. That's what we call fused or complete tetanus. We get one smooth, sustained contraction plateau, but that's also what leads to muscle fatigue because we're recruited and we're using all of our muscle fibers. So you can see all the stimuli being sent. One after another, after another, after another, after another, after another, after another, after another. They just keep coming. That is what causes this nice smooth contraction here. But then your muscles eventually fatigue out. And because we've used up all the ATP we have, we don't have access to any more enough ATP. And that's what causes muscle contractions to fail. And sometimes where your arms just kind of stop working for a few seconds or so, because then it allows your body to get everything back in order before you can do something again. So this is kind of what I've been talking about with that recruitment. So as you keep doing something, more muscle fibers are going to be sent, more stimuli. They're going to be more muscle fibers are going to be recruited to help you do something. That is technically called recruitment. So if we have a sub-threshold stimulus, let me draw my chart. Here my resting membrane potential. Here is threshold potential. A resting or a sub-threshold stimulus, we get a stimulus up, but it's not high enough to hit that threshold potential. So we don't see anything, nothing happens. A threshold stimulus. Well, something else comes. Oh, we hit that threshold. When we hit that threshold, our full depolarization and repolarization occurs. Maximal stimulus. That is, we get a strong stimulus to increase the maximum contractile force. So here's some pictures kind of showing all that. How many muscles are recruited for each? In the beginning of this, you can kind of see, oh, there's no stimuli that's going to produce a contraction. Because up here we see, nope, that's not threshold. Nope, that's not threshold. Oh, look, we just hit threshold. There was a contraction. But then these next one goes above threshold. Gives us another contraction. More muscle fibers recruited. Another stimulus. We go even higher. Another stimulus. More muscle fibers recruited. And as we keep getting more stimuli sent, when we hit that maximum stimulus, if you notice, all of these muscle fibers have been activated. So we get the maximum contraction we can have at this point. So how many muscle fibers get recruited? It kind of works on a size principle. Those smallest muscle fibers tend to get recruited first. Then as the motor units keep getting more stimulus, larger and larger fibers start to get recruited because they're getting more stimulus. We need more strength. The largest motor units, those are for like the most powerful contractions. Something that may happen because of an adrenaline rush, because something may have happened. That's when we need our maximum strength as fast as possible. That is, if you kind of think of it in that view, that is when that kind of happens. So those motor units, they tend to kind of contract asynchronously because it helps to fatigue. Because how I said, when you contract a muscle, some muscle fibers are contracting while others are not. We're trying to prevent fatigue for as long as we can in our muscles. So that's how our muscles, that's why as you train your muscles, it takes longer to fatigue out your muscles because only some muscles are getting recruited. Then if you keep doing the same movement over and over again, different muscle fibers will get recruited for the next time. Then different muscle fibers will get recruited to do the same action while the other ones relax again. We need that to help us be able to do something for longer periods of time. So here kind of shows the initial motor unit that is being stimulated. We get those small muscle fiber units first. Then more stimulus, medium muscle fibers usually come in. Then as we get more stimuli, that finally recruits those large muscle fiber units. We need something to sprint away from something. What is your muscle tone? What you see at somebody when you look at their muscles, when they're not contracting them, that is. If they're just sitting, the tone that you see in somebody's muscle is because our muscles are in a slight contracted state. They are constantly in this slightly contracted state. This is kind of what helps us. It keeps our muscles they kind of look good that way, but it keeps us healthy. It lets us respond to something fast. And it's because of spinal reflexes. Because of this, different motor units are alternately activated to give us this muscle tone. So let's look again. We mentioned isotonic and isometric contractions. Let's look a little bit more into these. So that isotonic and contraction, this is where the muscle changes in length. And that's when we can pick something up. They can be concentric or eccentric. A concentric contraction, your muscles are going to shorten and do work. Picking up your phone, that's a concentric contraction. Picking up your TV remote, picking up your computer, picking up a book, those are all concentric isotonic contractions. An eccentric contraction, that's when the muscle lengthens and generates a force. So when you've picked your phone up, you're doing something on your phone, that's that concentric contraction to pick it up. Eccentric is when you put your phone back down. Concentric, you're picking up a pencil or a pen. Eccentric, you're putting that pencil or pen back down. That is the difference between the two. Are you picking up something? That's concentric. Are you putting something down? Oh, that's eccentric. So here's those isotonic contractions. We have our concentric in this picture. We've got a 3-kilogram weight. In that concentric, we're going to pick up that 3-kilogram weight. But our isometric contraction, that load is greater than what you can pick up. So you're trying to pick up something that's like 150, 200 pounds. Most normal people cannot pick up something like that on their own. So that generates an isometric contraction. Your muscles are going to contract to try to pick it up. They're going to try to contract, I should say. They're not going to succeed because that muscle contracts, but it doesn't change length. Doesn't shorten. The muscle doesn't get longer. It kind of just stays the same. As I said, we're trying to pick up something that's 200 pounds. Your muscles are going to contract. They're going to try really hard to pick it up, but it can't do it. That is an isometric contraction. In that isometric contraction, actin filaments shorten. That causes some movement. Cross bridges try to generate the force, but those actin filaments do not shorten. Like those myosin heads, as your book says, they spin their wheels on the actin binding site. They try. They're trying to cock those heads to move. They just can't do it. So in an isometric contraction, our muscle here does not change length. That's what this means. So we've talked about ATP. Our muscles use that ATP to form those cross bridges, cock the heads of the myosin, and then the new ATP attaches to cause the myosin heads to release from those actin binding sites. So where do we get our ATP from? We have a store of ATP in our muscles, but we only have enough to last 4 to 6 seconds. So, and of course, our ATP is the only source of energy for contractions. So our body needs to be able to make it pretty quickly. But where do we use that ATP? As I said, it helps to detach those cross bridges from the actin and myosin. ATP pumps the calcium back into the sarcoplasmic reticulum. It'll pump sodium out of the cell and potassium back into the cell after the action potential has been sent. So we use a couple different ways to get our ATP. We can do direct phosphorylation of ADP by creatine phosphate. We'll look at that. We have an anaerobic pathway, which uses glycolysis, and it uses lactic acid to make energy. We have an aerobic pathway, which uses the electron transport chain to make ATP. So what is that direct phosphorylation of ADP by creatine phosphate? What exactly is it? So creatine, it's a molecule that is found in muscle fibers. That creatine phosphate is they give up their phosphate group so it can be combined with ADP to make ATP. It's done with the enzyme creatine kinase. That creatine kinase comes in, it reacts with that creatine phosphate, takes off the phosphate and attaches it to ADP to make ATP. So we have enough ATP and creatine phosphate to power a contraction for about 15 seconds. That's how much we have in our stores. We store this energy in our muscle cells. So they're what's usually used to begin a contraction. So here's this reaction. We have creatine phosphate and ADP. We use creatine kinase. That creatine kinase will take the phosphate off of the creatine phosphate, add it down to the ADP, and we're left with just creatine and ATP. We don't use oxygen for this, but we only get one ATP per ADP and creatine phosphate. Only one per molecules. So how do I know I talked about it and mentioned it a little bit in other previous chapters. This anaerobic pathway to make ATP. We use this process called glycolysis. It's a good 9, 10, maybe even 12-step process that works together to take glucose and turn glucose into 2 ATPs into pyruvic acid molecules is our other molecule that is besides our 2 ATPs. So each glucose gives us 2 ATP. So if you're doing something like sprinting across the street for something, you're using your anaerobic pathway because that's not something that's going to require high amounts of oxygen. So since there's not a lot of oxygen present, those low oxygen levels prevents the pyruvic acid from entering the aerobic respiration phase, or the electron transport chain, as it's called. Because pyruvic acid will enter the mitochondria and start aerobic respiration. But when there's a lot of at high intensity, so sprinting or lifting really heavy weights, oxygen is not available. If you're lifting some heavy weights, your muscles are contracting and bulging and they're kind of compressing blood vessels. So that's what causes oxygen to not be delivered as quickly to these muscles. So we use this anaerobic glycolysis to get 2 ATP, but then since that pyruvic acid is not being used in the mitochondria through aerobic respiration, it gets converted to what's called lactic acid. That lactic acid will eventually diffuse into the bloodstream. Because we talked about if we're lifting heavy weights, where muscles are contracting and they're blocking that bloodstream. So eventually when the blood stores kind of restored to the area, the lactic acid that was created through the anaerobic pathway will start to diffuse into the blood. It's also said that that lactic acid could be the cause of muscle soreness, because that lactic acid hasn't all diffused into the bloodstream. It's still stuck in some of those muscles. That is what is thought to cause muscle soreness. But anyways, that lactic acid, our liver, kidneys, and our heart can use lactic acid for energy because they take that lactic acid, they will convert it back to pyruvic acid or glucose in the liver. So we take that lactic acid in the liver and it can get converted back to pyruvic acid, or it can be converted back to glucose in the liver, which we can use to make energy again. Either one, because that pyruvic acid can enter the mitochondria of cells and go through the aerobic respiration, which aerobic uses oxygen, anaerobic does not. So anaerobic respiration here, it's only going to use about or create about 5% of the ATP we need. Because for each of these glucose molecules, we're only getting those 2 ATP. That's not a lot. 2 ATP, that's only going to disconnect 2 myosin heads from the actin. That's not a lot. So we need a lot more ATP than that. So let's kind of look. Here's that anaerobic pathway. We get glucose, which gets broken down from glycogen. Glycogen is energy stores in the liver. We use glycolysis, which creates 2 ATP. We don't use oxygen, so it gives us that pyruvic acid. That pyruvic acid gets converted to lactic acid. And this can give us about 30 to 40 seconds, maybe a little more of energy. Not much. But that aerobic respiration pathway, that can produce 95% of our ATP during rest. And it's also what is used when we do light to moderate exercise. It's slow, but we get more ATP from it. It takes those glucose molecules and it can break down to give us 32 ATP per one glucose molecule. So when we break down our glucose through the aerobic respiration, we get carbon dioxide, we get water, and we get 32 ATP. We can create a great deal of energy from this. But what if you're exercising longer than 30 minutes? If you'll exercise longer than 30 minutes, we start using our fatty acids as our fuel. So that's after 30 minutes of exercise. We start using those fats to generate energy, and that can produce a lot of energy as well. So here's our aerobic pathway. We have our glucose. It gets broken down from glycogen, which are usually stored in the liver. And when that glycogen gets broken down, it gets sent out into the bloodstream to get into the muscles. Or we can use pyruvic acid. It goes into the mitochondria. We use oxygen to convert either the glucose or the pyruvic acid into 32 ATP. We can use fatty acids as a source of energy, amino acids as a source of energy. But what we get out of it is, of course, carbon dioxide, our water, our 32 ATP, and we can generate energy for hours, which is a great thing. So just what I just said, aerobic endurance, doing, say you're running a marathon. You can do that for hours because you're using aerobic pathways to get energy. That anaerobic threshold, you're sprinting across the street. It doesn't require oxygen, so we use our anaerobic pathway, but we only get energy for a very short period of time. So here's the examples that I was suggesting. Sprinting, 30 to 40 seconds and you're out of energy. Running a marathon, running 100 miles. I've done it once. You can go for hours, even days, if you train your body right. You can go for days of running for a prolonged period of time. Kind of mentioned muscle fatigue. This is what happens when you reach that maximum contraction and you can no longer hold that contraction because there's no more ATP there. So that muscle fatigue is when you can't contract despite stimulation. We don't know the exact cause, but there's some possible causes of it. We can have ion imbalances, so potassium, sodium, calcium. We may just not have enough. We may not have enough, or we may have increased phosphate. And that ATP breakdown that kind of interferes with the calcium release. So it kind of hampers that power to have anything to happen. We get decreased ATP, and increased magnesium. Magnesium is known as a natural muscle relaxer. So if we get increased magnesium, it kind of interferes with those voltage sensitive T-tubule proteins, which causes that calcium to be released. But if we have more magnesium there, that calcium is not going to get released, which is going to cause your muscles to just not contract. Decreased glycogen. The lack of ATP is rarely the reason, unless you've severely stressed your muscles. And at that point, you're starting to look at injuries. So if you've ever exercised, run some distances before, you notice how after you finish exercising, you're still breathing heavier for a period of time. That is because we are trying to get enough oxygen back into our muscles to restore it to that pre-exercise state. So we need to replenish our oxygen. We need that lactic acid be converted back to pyruvic acid. We need our glycogen stores to be replaced. So we store our glycogen in our liver. We need to replenish those stores. We need to replenish that ATP and creatine phosphate reserves. We need to get that ready to go for the next time we do something. And they require oxygen. So that's why you're breathing heavier after you finish an exercise until you get back to that normal pre-exercise state. It is called excessive post-exercise oxygen consumption. It's formerly known as oxygen debt. To me, that's a little bit easier to remember than excessive post-exercise oxygen consumption. So that force of a contraction, that's all going to kind of depend on the number of cross bridges that are formed. And that's kind of affected by 4 factors. So how many muscle fibers are being stimulated? That has to do with recruitment. The more muscle units that are being recruited, the greater the force, as we've mentioned before. The size of the muscle fibers. The larger the muscle fibers, the more tension they can create. And if you do weightlifting, your muscle cells hypertrophy or get bigger. So as your muscle cells get bigger, they can lift more weight. They can generate more tension. They can create more force. The frequency of stimulation. Again, we talked about the more action potentials that are coming from that nerve being sent from the brain down to that muscle, the more action potentials being sent, the greater the force. And the degree of muscle stretch. When muscles are relaxed, Their muscle fibers at that sarcomere are 80 to 100% of their normal resting length. They generate more force. So if those muscle fibers are 80 to 100% of their normal resting length, they're going to generate a bigger force. If it's less than 80%, those filaments overlap too much, so the force decreases. There's not as much place for those actin filaments that are moving towards the center. And those actin filaments are getting pushed closer and closer by those myosin heads. They don't have as much room to go. So this contraction is not going to be as strong. If that sarcomere is greater than 120%, the filaments do not overlap. They do not. They don't overlap enough. So say that there's like one set of myosin heads that start the contraction. Then 2 sets can come in. Then 3 sets can come in. So the further apart they are, the less force they have, because maybe 2 myosin heads are working. Then 4 myosin heads are working. So these are some things that can increase the contractile force. How much frequency? How many frequencies or action potentials are being sent? How large is that muscle fiber? How many fibers are being recruited? How far is that sarcomere stretched out? So here's some examples. If we're at about 75% of our resting length here, you see how those actin filaments are overlapped? They're gonna not create that much force because they don't have a lot of room to go. If we are at exactly 100%, you see every one of these myosin heads here has a place to connect. So they're going to generate a very strong contraction. And this last one, the one I was just mentioning, there's only these 2 heads here and these 2 heads here that have a place to connect. So that's contraction is going to be very weak because there's only those 2 actin or those 2 myosin heads that are attaching to that actin to pull that actin closer for that first cross bridge. Then the second one. Then there'll be 4 on each side. Then there'll be 8 on each side until all of those myosin heads have a place to connect. Once they all have a place to connect, then they generate a stronger force. So how fast that muscle contracts and how long it keeps that contraction is due to the muscle fiber type. What kind of load are we lifting? And the recruitment. So how many muscle fibers are being asked to come join the party? So if we're looking at muscle fiber types, I'm not sure if you've heard of them or not, but we technically have what are called slow fibers and fast fibers. And they kind of relate to the speed of the contraction. Like how fast do myosin ATPases split ATP, and that pattern, of course, of action potentials being sent from that neuron. We have, and the metabolic pathway used for ATP synthesis. Are we using oxidative or glycolytic? Oxidative, oxygen. Glycolytic, glycogen. So that oxidative fibers are going to use those aerobic respiration pathways, which uses the electron transport chain. If you ever take biochemistry, you will learn all about it. We don't do it in here because it's taught in biochemistry specifically. Those glycolytic fibers, we use anaerobic glycolysis, and we use glucose as one of our energy sources. We get glucose from glycogen. So we have slow oxidative fibers, fast oxidative fibers, or fast glycolytic fibers. Our muscles are usually a mixture of the different fiber types. And that's what allows us to contract fast or allows us to hold contractions longer, because there's different fiber types within the same muscle groups. And that's what helps to prevent fatigue. But we talked about motor units. That motor unit is all the different fibers that one neuron talks to. They are all the same type of fibers. But in the entire muscle, like your biceps, you're going to have all three muscle type or fiber types in your, say, your biceps. Of course, your genetics are going to play a role in what kind of fibers, or what percentage of each fiber you have. Genetics plays a role in everything in the body, unfortunately, sometimes. So those slow oxidative fibers, they're slow to twitch, slow to contract. They use oxygen, so they're using that electron transport chain to gain energy. So if they're using oxidative pathways, it must be something kind of aerobic. Endurance activities, it's kind of low intensity. Even you say you don't do any kind of endurance activity, every single person does. Maintaining your posture when you're sitting in a chair, that is an endurance activity. So you can't tell me you don't do some kind of endurance activity. Everybody does it because your muscles are constantly working to hold your position, however you're sitting or standing. That is technically an endurance activity on your muscles. Those fast oxidative fibers, they're kind of medium intensity activities. You're sprinting or you're walking. They're needed a little bit quicker. They still use oxygen, so they're using the aerobic respiration pathways to get our ATP, but it's more medium activity, intensity kind of activities, even running long distances. That is it's a mix between slow and fast oxidative fibers that keep you doing something like that, something running for long distances. Fast glycolytic fibers, that's a very powerful, fast movement. Hitting a baseball, that is a very powerful, quick movement, and you're done. Hitting a golf ball, it's the same thing. Quick, powerful movement, you're done. So those use those fast glycolytic path fibers, which they contract really fast. They're using the anaerobic pathway to get your energy, and they're allowing you to finish whatever activity quickly. So let's look. Those fast glycolytic fibers, you're going to contract really fast. You are swinging a golf club. You're swinging a baseball bat. You're throwing a punch. That's a fast, hard. That would use your fast glycolytic fibers. Your slow oxidative fibers, they are also known as fatigue resistant fibers. They will help you do something for long periods of time. sitting in a chair for long periods of time. Those are your slow oxidative fibers. Running a marathon, those are slow oxidative fibers. So here's each of the different fiber types. This chart is direct from your book. And it talks about, say, speed of contraction, that myosin ATPase activity. So what that ATPase that converts ATP to ADP plus phosphate from those myosin heads, their pathway that they get their ATP from, their glycogen stores. Do they store any glycogen? What do they recruit? Their fatigue. Can they get fatigued fast or slow? Some examples. Those slow oxidative fibers. You're running marathons, maintaining your posture. So your muscles contract when there's no load added. So you bend your arm up, there's nothing in your hand. Your muscles are going to contract really fast because there's no extra weight added on. The greater that load, so the heavier the object you're trying to move, the slower you will contract those muscles. But the more motor units that are being recruited to this contraction, the faster the contraction will happen. 1 motor unit versus, say, 100 motor units. 100 motor units is going to contract a lot faster and it's going to hold a lot longer than, say, one motor unit, one set of muscle fibers. So here are some charts that shows we get a stimulus, a light load, oh, that contracts the fastest. And medium size load, that contracts a little bit slower, but that heavy load, it contracts a lot slower. So different endurance activities. Aerobic exercise, like jogging, swimming, biking. What that does to your body is it produces more capillaries in your muscles because you need more blood supply. You need more oxygen, more nutrients going to your muscles to exercise. We get more mitochondria in those muscles because we need more ways to generate ATP. We need to get as much more ATP for when we're exercising. We get more myoglobin synthesis in our muscles. That helps to kind of store our oxygen. And as we gain more of those, you get more endurance. That's why marathon runners, they start running short distances, like 3 or 4 miles. I know that doesn't sound short, but they usually start their training at like 3 or 4 miles. Then over the course of about 3 to 4 months, they're slowly increasing their mileage until their final run before their marathon is usually about 20 miles. Then they go run their marathon, which is 26.2 miles. What their body is doing as they're training for these marathons, they're getting more capillaries as they continue the training. They're getting more mitochondria for more energy. They're getting more myoglobin. They're helping to create what's needed so they can go further distances to help resist fatigue. Because of this endurance aerobic type activity, we can convert fast glycolytic fibers to fast oxidative fibers to help contract as we continue running. It's kind of cool. Our body can kind of change things up a little bit to work with what we're trying to do. Resistance exercise. That's weightlifting usually. And that causes our muscles to hypertrophy or get bigger. And that is because of increased fiber size. As the muscle fiber size increases, our muscles get bigger. But we also increase our mitochondria. We get more myofilaments, we get more glycogen stores, which also leads to more connective tissue. That'll help our muscles get stronger and bigger. Our muscles must remain active to be healthy, including just walking. That is a way for our muscles to maintain some sort of health. We have what is called disuse atrophy. It's exactly what it sounds like. Disuse, you're not using your muscles. So if you're not using your muscles, they're going to atrophy or get smaller. They're going to lose mass. Somebody who is bedridden, they're going to have smaller muscles. If you've ever had a cast on an arm or a leg somewhere, when they take the cast off, the leg that had the cast on is a lot smaller than the leg that did not, because that leg was not being used. So those muscles, they lost their muscle mass. Here's the sad part. The strength can decline by 5% per day. So if something were to happen and your muscles become paralyzed, they can atrophy to 1/4 of their normal or their initial size. And if you continue to not use your muscle, somebody who's bedridden, fibrous connective tissue starts to replace muscle tissue. And once this happens, this fibrous connective tissue replaces muscle tissue, we can't rehab that. That's done. That's beyond the point of repair. So that's why when there's people that are bedridden in nursing homes, they usually have somebody to come in and at least help them move their arms and their legs to help get muscles contracting, muscles moving, to try to prevent this disuse atrophy. Because they're trying to prevent that fibrous, that connective tissue replacing their muscle tissue, because that's beyond the point of repair. We can't do anything to rehab that anymore. So now that we've spent over 100 slides talking about muscle, skeletal muscle, let's look at smooth muscle. As we stated at the beginning of part 1, our smooth muscle is found in the walls of most hollow organs, like our respiratory system, digestive system, urinary system, reproductive system, circulatory system, except the small blood vessels. And we don't have smooth muscle in our heart. Smooth muscle is usually arranged into what kind of like sheets. They're, remember, they're those fusiform type fibers. They kind of fit together really well. They kind of form these little sheets. So Because the smooth muscle is in sheets, most organs tend to have two layers of these sheets, and they're kind of oriented at like a right angle to each other. Right angle is at about 90 degrees to each other. And that helps to provide some of that strength that is needed to help hold, say, your stomach, your uterus, especially in females that are carrying a baby in there. We need that muscle strength. So we have what is called a longitudinal layer and a circular layer. So that longitudinal layer, fibers run more this direction. Well, that circular layer, fibers are going to run around in this direction. Because of the different layers going different directions, they can alternate their contractions and relaxations, because that's what helps move things along. That helps mix things up in your stomach, per se. That's what helps move things along in your intestines. I talked about there's been kind of a fusiform spindle shape kind of fiber. They're those long, thin, or not long, they're thin and short compared to skeletal muscle. They have one nucleus and there's no striations. They do not have those connective tissue sheaths. They only have that endomyosin. They have what are called varicosities, or these bulbous swellings of nerve fibers, instead of that neuromuscular junction. They store, those varicosities will store and release neurotransmitters into that synaptic cleft when they need to contract for something. And that is, in smooth muscle, it's called a diffuse junction. Smooth muscle, of course, is innervated by our autonomic nervous system because we do not have control over it. So let's look at how the two skeletal muscle versus smooth muscle, how are they different? They don't have as many thick filaments, but they have myosin heads that run the entire length of that cell. Their thick to thin filaments is 1 to 13. Skeletal muscle is 1 to 2. Remember, skeletal muscle has the thin filament, a thin filament, and that thick filament between the two thin filaments. Thin filament, or smooth muscle, does not have that. But those thick filaments, they have those myosin heads along that their entire length. That's what kind of creates smooth contractions for the smooth muscle. They do not have they do not have that troponin complex. They do have tropomyosin, but they have calmodulin that binds calcium. Smooth muscle has calmodulin that binds calcium. So here are these varicosities. These are the nerves from the autonomic nervous system. Each one of these little heads here is going to attach to a different muscle cell. So they're going to tell these muscle cells to all contract together. Smooth muscle doesn't have the same elaborate sarcoplasmic reticulum. They do not have T-tubules. But just like skeletal muscle, they still have calcium in their sarcoplasmic reticulum. But they don't really use that calcium for contractions. They use the calcium that's in their extracellular matrix. Their sarcolemma have caveolae, which are these little infoldings, these pouch-like infoldings. And that's where the calcium channels open and allow calcium into the cells. So here's a smooth muscle cell. We have these calcium channels. These are the calveolae, these little kind of invaginations here. That is a calveolae. Calcium is going to enter that calcium channel in this calveolae here. That calcium comes from that extracellular fluid into the cell. That calcium that comes in triggers calcium to be released from that sarcoplasmic reticulum. So calcium is coming from two directions. Calcium is coming in this way, and it's coming out of that sarcoplasmic reticulum this way. So we're getting calcium into that cytoplasm from two different directions. Just like skeletal muscle, smooth muscle is electrically connected. They have gap junctions. But skeletal muscle, they're electrically isolated. So when that smooth muscle contracts, those gap junctions allow that depolarization from one cell to spread to the next cell, to the next cell, to the next cell, and to the next cell. They do not have striations and they do not have sarcomeres, but they do have thick and thin filaments. So here is a smooth muscle cell. See all these little kind of white things in here that I'm coloring yellow? Those are those calveolae, those little invaginations into that smooth muscle cell. That is where those calcium channels sit. These gap junctions right here, these are what connect one smooth muscle cell to the next smooth muscle cell. This allows an action potential that was sent to this fiber to spread to the next fiber through that gap junction. Unlike skeletal muscle, those thick and thin filaments are more in a diagonal shape because those smooth muscle cells don't contract straight across like a skeletal muscle does, fiber cell does. They're going to kind of corkscrew, they're going to spin when they contract. And that's what that diagonal thick and thin filament is going to help to do. They have intermediate filaments that kind of create this a lattice-like arrangement of non-contractile filaments that help to kind of resist too much tension. They have dense bodies. Those help to anchor those filaments to that sarcolemma. They have Z discs. So during that contraction, that sarcolemma actually starts to bulge outward. It makes that muscle cell look puffy. So here's a contracted smooth muscle fiber. You see these dense bodies here that kind of help with this smooth muscle. This muscle cell has contracted. It kind of went in this corkscrew contraction. And as it contracts, it makes this muscle cell look a little puffy. This is what we were just talking about on the previous slide. So here is that table from the beginning that shows skeletal muscle when we've looked at smooth muscle. So it kind of talks about, it shows histologically what they look like, what they do, where they're located, cell shape, different kinds of fibers, how they may have sarcomeres versus T-tubules. Here is how they communicate with the nervous system, how they communicate with each other. They're thick and they're calcium. Where does calcium attach? What kinds of contractions they create? So our smooth muscle, they tend to create a more slow, synchronized contraction. So if one cell gets stimulated through an action potential, that gap junction is going to send the information from one cell to the next cell. And that allows those action potentials to get transferred from fiber to fiber. We have some smooth muscle cells that are self-excitatory. They don't need an action potential to contract. So they are considered pacemakers. So what they do is they make sure, even if we're not getting stimulation from a nerve to contract, they make sure that smooth muscle still contracts to move stuff through your intestines, to move stuff out of your stomach. These pacemaker cells make sure that your body, your smooth muscle is doing what it's supposed to do. The rate of contraction and that intensity, that is modified by your nervous system and through the chemical connection, talking back and forth between that neuron and that cell through neurotransmitters and different ions. And those contractions in the smooth muscle, they're still pretty similar to skeletal muscle because they still do that sliding filament with the actin and the myosin. That intercellular calcium level determines if a contraction is going to happen. We still use ATP to move those myosin heads to slide that actin along. And when calcium's no longer available, that contraction stops, just like in skeletal muscle. Those are all the same. But how is it different? How are these contractions different? We get some calcium from the smooth endoplasmic reticulum and smooth muscle, but most of it's going to come from that extracellular space. That's where we get that calcium from. Calcium binds to calmodulin in smooth muscle. And that calmodulin activates the myosin kinase. And that myosin kinase will phosphorylate the myosin head. That leads to that cross bridge formation. But what happens to stop a smooth muscle contraction? It's pretty easy in a skeletal muscle. Calcium gets stored back into that sarcoplasmic reticulum. There's no calcium to bind to the troponin that removes that tropomyosin string on the actin heads that allows the myosin heads to attach and cock and move that actin. And as soon as there's that calcium is released, that tropomyosin string gets put back over that binding site in skeletal muscle, so skeletal muscle can no longer contract. Well, what happens in smooth muscle? Well, that calcium has to detach from the calmodulin. Calcium gets put back into that sarcoplasmic reticulum or extracellularly, so it's kicked back out of the cell. We also have to dephosphorylate the myosin into an inactive form. So here's all these steps. So we see calcium ions come in to the cell. Calcium is going to bind to this calmodulin here to get an active calmodulin. This activated calmodulin that activates the myosin on that the light chain kinase enzyme. So we get that inactive kinase to that active kinase because of that calmodulin. That activated kinase enzyme transfers the phosphate to the myosin. That's how we go from ATP to ADP plus phosphate. Then those myosin heads will bind to the actin and cause the muscle to contract. Our smooth muscle is a lot more energy efficient than our skeletal muscle because they contract slower and they relax. But they can maintain contractions for a long time and they don't use a lot of energy to do so. Their ATPase is a lot slower to hydrolyze that ATP. We have myofilaments that may latch together to save energy. So most of our smooth muscle can maintain some kind of contraction, just like our skeletal muscle, because that keeps our muscle tone. And that is, of course, revert to our smooth muscle tone. We have our skeletal muscle tone and we have our smooth muscle tone. We make ATP in smooth muscle via the aerobic pathways, that aerobic respiration. So our nerves, our hormones, and different chemical changes around those cells are what regulates the contraction of a smooth muscle. We have our neurotransmitter that's going to bind, that's going to cause the start of an action potential. And that's what causes, again, our calcium to come into our sarcoplasmic reticulum, which we've kind of talked about. But that all depends on, is neurotransmitter released? And what type of receptor molecule do we have? One neurotransmitter can stimulate that smooth muscle, but it have an inhibitory effect on a different organ. So our neurotransmitters do a lot of different things. There's a lot of different neurotransmitters. When we get to the nervous system, we'll look into some of those in some more detail. But we have one neurotransmitter. It can cause one, like the smooth muscle in your stomach to contract. But if that neurotransmitter gets released, say at your arteries, your larger arteries to contract, that same neurotransmitter there may prevent contractions from occurring. Some smooth muscles don't have any nerve supply. That's where we have those pacemaker cells because they depolarize spontaneously. Those pacemaker cells are going to allow those muscle cells to contract and spread those contractions from cell to cell to cell. Or they could depolarize because of a chemical stimuli. Like our hormones. Our hormones do all kinds of things. If we have too high of carbon dioxide in our body, the pH, we don't have enough oxygen. That can cause smooth muscle to contract or not contract. Some of our smooth muscles can respond to both neural and chemical stimuli. So our smooth muscle has a stress relaxation response. They can respond to stretch, then adapt to new length. But they retain that ability to contract again. Think of your urinary bladder and your stomach. It allows you to put contents in there, food into your stomach, which will cause your stomach to expand. Your urinary bladders, your kidney is filtering your blood. Any of the waste products get stored down into the bladder to be released. And that allows your bladder to enlarge as more urine gets stored in there and eventually gets released. Length and tension changes. They can contract between half and twice its resting length. So that allows our organs to contract, but they don't become flabby. That's the last thing we want, is our stomach to become flabby. That allows our uterus, after giving birth, to go back to its regular size. Because it'll go back to its regular length. Smooth muscle, they differ from organ to organ because different organs have different needs. Some of them may have different fiber arrangements. Some of them have different innervations. Some of them respond to different stimuli. So they're either unitary or multi-unit. A unitary smooth muscle or a visceral muscle, it is found in all hollow organs except for the heart. That is where we have that longitudinal sheets of cells and then those circular sheets of cells. They have those varicosities. So all those different, they all kind of attach together. They come out to one neuron. They can create spontaneous action potentials, meaning they can contract without the nerve sending information. They have gap junctions. So one cell gets stimulated with an action potential and it can send that information to every cell surrounding it. And they respond to different chemical stimuli in a unitary smooth muscle. In a multi-unit smooth muscle, that's kind of in the large airways of our lungs, large arteries, erector pili muscles in your arm, allows your hairs to stand up. The iris of your eye. There's not a lot of gap junctions, and they don't really depolarize. They don't have those pacemaker cells. So they're pretty similar to skeletal muscle. They're independent muscle fibers. They have that autonomic nervous system innervation, and they form motor units just like skeletal muscle does. They have graded contractions like skeletal muscle because they're responding to the amount of stimuli. But they're different from skeletal muscle because it is controlled by the autonomic nervous system and hormones, because our hormones can tell smooth muscle to do something. So let's quickly look at how our muscles develop. All of our muscles come from myoblasts, which is an embryonic cell. Our skeletal muscles are all formed by fusion of many different myoblasts together. That's what gives us our cells those multinucleated appearance. We have a growth factor, and that's what kind of stimulates those acetylcholine receptors at that neuromuscular junction that we went through in part 1. Those cardiac cells start pumping at 3 weeks old. They start contracting and pumping. When you get to the cardiology unit in anatomy 2, you learn how they contract. So here's how they start. They start as these little embryonic cells. Mesoderm cells. They turn into myoblasts. Many myoblasts come together, they fuse together to make a myotube that turns into a mature skeletal muscle cell. Satellite cells are also known as stem cells, kind of for muscle, but they differentiate into muscle cells. So, as I just said, those satellite cells are kind of like those stem cells. They have a limited regenerative ability. They don't regenerate very well. They can, but not very well. Cardiomyocytes can divide at a modest rate, but say you get a heart attack. A heart attack is when one of the blood vessels that goes to the heart gets blocked, so blood is not going to the heart muscle that allows that heart muscle to contract. So what happens is that those cells around that blood vessel they're not getting any nutrients anymore, they die and they get replaced with connective tissue. Your smooth muscle, that regenerates through life. Out of your muscle tissues, your smooth muscle regenerates the best. Cardiac and skeletal muscle can lengthen and thicken, and that can lead to hypertrophy or larger muscles. So in infants, as our muscles develop, it helps to kind of create that neuromuscular coordination. You ever notice infants aren't always the most coordinated? As they start moving around, it starts to help train the brain on how to make these muscles move correctly. So these developments happen from head to toe, proximal to distal. Just like a baby can lift its head before it is able to walk. That's an example because they start with their head and they move to their feet. We have our peak neural control by mid-adolescence. With athletics and training can continue to improve your neuromuscular control. The longer you do exercise, it can keep your neuromuscular control in peak condition a lot longer. Females, their skeletal muscle is about 36% of their body mass, while males, it's up to 42%. And that's because of their testosterone. Testosterone allows for males, their muscles to get bigger. So I said they have a large ability to enlarge their muscle fibers because of that testosterone. But body strength per unit mass is the same for men and women. So what happens when we get old? everything kind of goes downhill as we get older. So let's see what happens to our muscles. So as we get older, we get more connective tissue within our muscles. Our muscle fibers start to decrease. By 30, we can lose muscle mass. That's when that starts, at 30 years old. Sarcopenia is losing muscle mass. So if you exercise regularly, you can reverse that loss in muscle mass. Atherosclerosis, that can start to form. That is where in blood vessels, here's a blood vessel, we get a fatty plaque buildup. It starts to block blood vessels. It can lead to what is called intermittent claudication. And that's where blood is not flowing very smoothly. And it can cause severe leg pain. Another reason to exercise. We talked about exercise with our bones. Now we talked about how exercise can help us with our muscles. So thank you for listening. Goodbye.